Now we take some time and we look at the pieces in more detail. We've seen these pieces all semester so far. We've talked about them before, but now we're going to look at them in a little more detail in regard to how they fit into the immune response against parasites, but also associated with antigens or with allergies. So IgE. IgE is an antibody that we've looked at in the past. It has the epsilon constant region, and really its role in the immune response or in the immune system is to work with mast cells, eosinophils and basophils, by attaching to their surface and being a surface receptor. And when the appropriate antigen engages IgE, then signals will be sent down through those cells to tell them to degranulate. Now, the appropriate antigen that should be recognized by IgE should be some sort of parasite, maybe a multicellular worm. It could be maybe some other parasites, but those are the appropriate antigens. And there's multiple epitopes on, let's say, a multicellular worm or helminth that can engage multiple antigen receptors. And that would lead to cross-linking, which allows for the signal to be sent down through the cell. And so these parasite-specific IgE molecules get sucked up as soon as they're produced by a plasma cell and attached to the surface of these cells. These eosinophils, basophils, and mast cells have what are called Fc epsilon receptors. And so here is our Fc epsilon receptor. And what that does is it recognizes the Fc portion of the epsilon heavy chain. And so then those IgE will bind so that their antigen recognition portions are sticking out. Once the antigen receptors are engaged by the parasite, then there's going to be that cross-linkage that occurs, and there will be a signal sent down to the cell, which will then allow these cells to activate release their inflammatory mediators out into the extracellular space. And then that's going to lead to inflammatory responses that hopefully will eliminate the pathogen or eliminate that parasite or lessen the parasitic load. So helminths, they are worms. It's another name for them. And they are the most widespread multicellular parasite in humans. These include things like flatworms, the tapeworms, roundworms like hookworms, and maybe some other roundworm worms. Could be some nematodes, but those are mostly dirt. But anyway, roundworms. And then the flukes. The flukes tend to live in certain organs like the liver, lungs. You can have, those tend to be the same. Blood can live in the blood as well. Anyway, these worms, they can live there and they can, be inside of somebody and somebody can be okay with it, but they hang out for a long time and they can cause chronic illness. It can be debilitating. This should say hosts, not hoses. Sorry about that. But they compete. They end up stealing food from their host and nutrients, and there could be tissue damage along the way. So depending on what type of fluke or not fluke, but depending on what type of worm you're looking at, there's different pathologies that go along with that. Now, parasites, because they're so large, they're not easily killed by macrophages or neutrophils. Those single cells, although macrophages are pretty large themselves, can't ingest an entire helminth. And so the strategy that the immune system uses then is rather than ingesting them, they will just dump granules onto the surface of the parasite or onto the surface of the worm, or utilizing the body's force to eject the parasites from the body. This can include things like coughing, sneezing, blowing your nose, throwing up, having diarrhea, maybe an itch, scratching to break the skin to allow the organisms to get out. These are all ways that those mast cells, eosinophils and basophils, physiologically get the body to do these types of activities to remove the parasites. And so this expulsion of helminths from the GI tract is really a coordinated set of events that utilizes both innate and secondary immune responses and cells, as well as muscle cells of the body to coordinate like explosive diarrhea. It takes a lot of cells to get that to happen. And this is also true for sneezing and coughing and things like that. But it is a little bit different approach rather than just phagocytizing these types of pathogens. They have to be removed in different ways. IgE is central to this orchestrated event, this response. And because it's IgE driven, we call it a humoral response. It is a humoral response, and it's going to be polarized towards the T helper 2 side, because the T helper 2 cells, those effector T cells, are going to secrete cytokines that will drive the isotype switching of B cells to IgE, and will also help with not having such an inflammatory environment. So the T helper 2 is the polarized pathway best for eliminating parasites. And these parasite-specific IgE antibodies are used to arm the basophils, the eosinophils, the mast cells, and really then end up being the receptors for these cells so that they can find, locate, and attack the parasites. So humoral response is going to be a beneficial response towards a parasitic infection. An inflammatory response that we see with the T helper 1 is not ideal for parasites because interferon gamma can be very destructive. High inflammation isn't going to help eliminate the parasite in the long run, but it can actually cause more tissue damage, disease. This should say severe. I did not do a very good job editing my PowerPoint. Anyway, so T helper 1 would actually exacerbate the destruction of tissue and not even clear the parasite anyways. So it is a much better approach for the body to drive a T helper 2 response for parasites. And these are going to be cytokines like IL-4, IL-5. So an abundance of mast cells. Remember, mast cells are going to be found in the tissue. They're not going to be circulated in the blood very much or anywhere else, but they're going to be in the tissue itself. And then when these parasites infect that tissue, it's going to be important for those mast cells to have IgE on their surface so that they can provide a protective TH2 response. So these high affinity Fc epsilon receptors on these mast cells are going to bind up IgE as soon as they can find it and hold on to that IgE and use them as receptors. So even when there's not antigen present, mast cells are armed with antigen-specific IgE just waiting. And these antigen-specific IgE do not have to be consistent across the cell. They can be many different types of variable regions because the Fc epsilon receptor is only recognizing the Fc portion. If a helminth antigen then binds to an IgE on a mast cell, cross-linking can occur, and then the signals get sent to the mast cell to degranulate. Those granules are going to be filled with very inflammatory mediators like histamine, not like what we see with interferon gamma, but rather histamine, which is going to have a physiological effect of making the tight junctions leaky and allowing fluids to flow, allowing for Yeah, fluids to flow, which if it's in the intestines, it can lead to diarrhea, runny diarrhea, and muscle spasms, and things that you would characterize with explosive or very forceful removal of things in the gut. And hopefully that will pull out the worms and make the worms go away as well. That's the goal. Now, the T helper 2 cells, again, on the humoral side of the response, yes, we have IgE antibody being produced, but IL-5 is a cytokine that is going to not only help with class switching in B cells, but will also drive eosinophil production and control their development. And when we have a helminth infection, then the presence of IL-5 is going to ramp up the number of eosinophils that are present. Eosinophils live in the blood. That's typically where they're going to be found. They can also be found in other tissues, nasal, upper respiratory for sure, and then maybe in the gut. But wherever these helminths are found, IL-5 will increase the production of eosinophils, but then draw the eosinophils to that infected area. And like mast cells, eosinophils also have the Fc epsilon receptors. And so they will be bound with IgE, and they also can recognize the epitopes on the helminth. Cross-linking occurs and allows degranulation of eosinophils. So very similar process. But even I love this picture. I think what makes me love this picture so much is the detail that goes into these granules that are being dumped directly on the surface of the helminth. This is the worm here, like a little smiley face, right? This worm, although it should be a sad face because this worm is getting burned. Literally burned. These granules look super painful, and they're falling onto the surface of the helminth and causing destruction to the helminth itself. So not only do we have mast cells that are forcefully trying to get these parasites out of the gut, in this case, but we're also having eosinophils dumping granules. It's like a 1-2 punch to these helminths. And so these are toxic molecules directly affecting the helminth survival. And then you have the mast cells secreting histamine to cause diarrhea and muscle contractions to get the worms out as well. So it's a pretty good strategy to remove parasites if there's parasites present. Right. So what we have now is in industrialized countries in the world, the human parasites are not very often found because they have really been eradicated or at least drastically reduced because of sanitation protocols. And so the same mechanisms of recognition and effector function that would normally be utilized against helminths, because there's no helminths there, they kind of got to be used somewhere. And so exposure to antigens then that might not be too big of a threat can actually cause the IgE molecules to be engaged and cause what we would want to have happen for helminths happen to things that really don't cause a problem, like grass or pollen, things that really are just allergens. And so in this situation, IgE, mast cells, basophils, eosinophils, I don't have that on here, actually, instead of removing a helminth, they just end up causing what we know as allergy. And so you get immunopathy, pathology, destruction of tissue for antigens that really don't matter. They're not going to pose a threat to human health. And so this is why we see allergies in places where we don't have a lot of parasitic infections. So here's a map of the world where we see, at the time of the publishing of this text, where we see a lot of higher incidence of parasitic infections. And you can kind of line it up with, yes, the tropics, because you have a warmer climate, but these are also going to be in areas that are not as economically industrialized. And so you don't have a very high, widespread sanitation of water, of just cleanliness of all kinds of things. So parasitic infections are very endemic in these tropical countries. And many people in the world, like 1.5 or more, 2 billion people are infected with worms. And we don't see allergies in these countries. These countries don't have the epidemic of allergy that we see in the more industrialized or the northern countries and southern countries outside of the tropics. So this brings up this interesting hypothesis called the hygiene hypothesis. Maybe you've heard of it before, but here are the big things that go along with the hygiene hypothesis. We like to be clean, right? And so excessive hygiene, and especially around children, we like to clean kids' hands, and babies, we like to wash our hands before we touch them because we don't want them to get sick. And that makes sense. That makes a lot of sense. But by reducing babies and young children to the exposure of worms, maybe let them eat dirt. If they don't eat dirt, they're probably not going to get a lot of worms, or if they're not, whatever. Cleaning them removes a lot of the exposure they may have to helminths or to other parasitic microorganisms. We vaccinate, and there's vaccination all over the world. We have programs all over the world, but highly well-distributed vaccination programs in industrialized countries. And so they're not facing natural infections that we would have seen 100 years ago or 50 years ago. And we use a lot of antibiotics, and this over-reliance on antibiotics to terminate infections and not let the immune system become engaged. All of these things together really culminate in that childhood development of immunity is not as robust as it has been in the past. And so the development of the immune system is held back. And we know that with research with mice that are never exposed to pathogens, their immune systems don't develop. And so we know that immune systems need to be challenged in order to become fully developed. But if we're constantly removing these things that can develop the immune system, we can end up with a dysfunctional immune system. We could end up with maybe children reacting to things that aren't necessarily going to be a threat, like allergens. And so those are the tenets of the hygiene hypothesis. Okay, I want to kind of just wrap up here with another set of cells, the mast cells, and then we'll look at the eosinophils as well. Mast cells are found in the tissues. They are saturated with IgE because they have those epsilon receptors on their surface. And they are going to, again, have a variety of different antigen specificities because they could have a variety of different types of IgE on their surface. And in the absence of antigen, mast cells are going to have their granules. They make those granules, they protect them in little vesicles, and they hold on to them, and they don't do anything with them until those IgEs are cross-linked. And so then when antigen is encountered, it has to be the second time, the secondary immune response, then because those IgE wouldn't exist until the secondary immune response. When antigen then is encountered, then those IgEs can be engaged and tell the mast cell, okay, release your inflammatory mediators. When that happens, then this then allows for the release of what was those granules that were stored inside of the vesicles, and they are going to be chemically they're going to chemically change the zone in which they're released, and it's going to create leukocytosis. It's going to so bringing in more white blood cells, and this is going to benefit if there's a pathogen. We want this to happen in the case of maybe some sort of parasite. But if this cross-linking happens because of an allergen, there's just a lot of destruction that doesn't need to be. And so there's damages to the body and tissue, there's dysfunction, there's all kinds of different side effects for no reason, because those pollen grains, for example, like in this picture, pose no threat to the health of the individual, where a helminth could pose threat. So that's what's happening with mast cells. We're going to see similar things with eosinophils and basophils. Eosinophils are part of the granulocyte flow chart, right? If you look at the hematopoiesis, we have the myelocytic lineage, and then some of those are granulocytes, and one is eosinophils, one is basophils, and then basophils is kind of suspected that once basophils move into the tissues, they become mast cells, because basophils and mast cells look very, very similar in their properties. But anyways, eosinophils are granulocytes. They're usually going to be in the tissues. They're going to be in the blood, but in the tissues, like in the connective tissue, like in the upper respiratory tract, right below the epithelial lining. They're going to be there ready and waiting for any helminths or any parasites that might come in. And this is true of the respiratory tract, but also the GI tract, urogenital tract, any of those mucosal associated tissues. Then the activation of eosinophils is similar to what we saw with mast cells. There's going to be IgE on their surface. They need to be cross-linked in order for those granules to be released. They're going to have inflammatory mediators, and in the normal function, they're going to be really good at killing parasites by dumping the granules right on there. In an allergic function, they're really good at making you sneeze and have runny nose and eyes. Yeah. Okay, and basophils. This is the last little bit. Again, one of the granulocytes. Remember, they're neutrophils, eosinophils, basophils are the three granulocytes. They have similar set of mediators like we saw in mast cells, and they also will bind up IgE because they have the epsilon receptors. Now there's some talk about how they can bind IgD, but nobody really cares, at least at this point. We don't really know about IgD, okay? Anyway, mast cells, eosinophils, and basophils usually all act in concert and lead to what we see happen for expulsion or killing of parasites, or on the flip side, an allergic response if there's no parasites present. So now you can see, you probably have a better grasp on why we connect allergy with parasitic response in this chapter.